Okay, hey guys, this is Mrs. Kraus, and this is our second video on the Unit 2 Part 3 notes on macromolecules. At this point, we've talked about three of the macromolecules, carbs, lipids, and nucleic acids, and we're about to go on to the fourth macromolecule, which is proteins. Proteins contain the elements carbon, hydrogen, nitrogen, oxygen, and sometimes they contain sulfur as well. Proteins are known as the workhorses of the cell because they have many different potential functions. Some of those functions are listed here. They could be used for structure. For example, the proteins in our hair and nails provide structure. The specific protein in your hair and nails is keratin. It's the same one that's found in like cow hooves. Gross. We can also use proteins for transport. For example, there's a protein called hemoglobin in your red blood cells that binds to oxygen and carries oxygen through the bloodstream. We also use proteins for movement. You have proteins in your muscle that help contract the muscle to allow you to move. We also use proteins in the immune system to defend our body against pathogens like bacteria and viruses. And those specific proteins that assist with defense are called antibodies. Proteins are also used to control signals sent between cells and also reactions inside cells. So controlling signals between cells, those are hormone proteins, and then assisting with reactions inside cells, those are proteins called enzymes. The monomer or single building block for protein molecules are called amino acids. We have 20 different types of amino acids found in nature, and each amino acid has the same basic structure. Each amino acid has a central carbon atom, which is pictured right here, that's bonded to four things: a single hydrogen atom, right here, an amino group, right here, and the amino group always contains nitrogen. It also has a carboxyl group. A carboxyl group always contains a carbon double bonded to an oxygen. And then the last thing is an R group. The R group is also called a variable group, which means it's different in every amino acid. So that's the thing that differs between our 20 types of amino acids. Okay. Proteins actually consists of polymers called polypeptides. A single polypeptide is one chain of amino acids linked together, and usually a full protein is going to be several polypeptides that are folded around each other. The specific folding pattern gives the protein its shape, and that determines the protein's function. I'm going to give an example. Hemoglobin actually has 4 polypeptides that are folded around each other to make that full hemoglobin protein. Now, within proteins, we have 4 different levels of shape or structure: primary, secondary, tertiary, and quaternary structure. And they work together to give the protein its ultimate shape, which determines its function. Okay, so I'm going to get kind of in detail about the different levels of protein structure. The first level, the primary level, we can write primary with a 1 and a little 0 superscript. The primary level of structure is just covalent bonds that form between neighboring amino acids within a single polypeptide. So remember, a single polypeptide is going to be multiple amino acids or monomers joined together in a chain. So that's just the order of amino acids, which determines our primary structure. Secondary structure is when hydrogen bonds form between the amino and carboxyl groups on amino acids that are not next to each other within a single polypeptide. So remember, the amino group has a nitrogen in it. The carboxyl group has a carbon double bonded to an oxygen. So within the secondary structure, hydrogen bonds are forming between those two groups on different amino acids located in different parts of the polypeptide. That results in 2 different shapes forming: either an alpha helix, which looks like a spiral, and you can see the little hydrogen bonds that are forming, or it might also result in a beta pleated sheet, which looks like if you were to fold a piece of paper back and forth, and then it looks like an accordion when you open it again. Okay. The next level of protein structure is the tertiary level. This is when proteins start to get really different from one another because all of them have some alpha helices and some beta pleated sheets. But at the tertiary level, we see different types of bonds like ionic, covalent, or hydrogen bonds forming between R groups. And remember, R groups are different for all the amino acids. So depending on which amino acids you have in your polypeptide, you're going to have lots of different interactions between the R groups on amino acids within that polypeptide. So we see lots of different crazy structures forming at the tertiary level. At this point, we're still within a single chain of amino acids, a single polypeptide. The quaternary level is when you have different polypeptides that fold around each other. So that's why I've shown different colors here. We have a blue polypeptide, a red one, and a green one, and they're folded around each other. What forms the quaternary structure is when different types of bonds like ionic, covalent, or hydrogen occur between R groups of amino acids on different polypeptides. So the R groups of amino acids on the blue one are bonding with R groups and the amino acids on the green one. That results in your multiple polypeptides folding around each other to give you the full protein, and our final shape determines our function. For example, antibodies, which are used in your immune system, are usually shaped like a Y. The reason they're shaped like a Y is the branches of the Y are able to latch on to a bacteria or virus, and they're able to hang on and actually mark that bacteria or virus for destruction by the rest of the cells in your immune system. Crazy. So the antibody protein is shaped like a Y so that it can latch on to bacteria and viruses and mark them for destruction. Go figure. All right, that's it for macromolecules. Thank you.